on as well.
Yeah.
Okay. It's a poor showing, Kate, but you're awake. We might as well crack on.
No worries.
Thanks for coming. Thanks for doing this talk again. I always look forward to it, even if I should know most of the stuff, but I don't pay as much attention. At least I have the excuse now that I haven't been talking to you in a few months, so it's not that I've just been ignoring all your emails. Yeah, thanks for doing the talk, and looking forward to what should be a great talk on kelp ecosystems in Ireland.
Yeah. Okay. Well, I'll just kind of jump into it, and then I'll try and keep an eye on other attendees trying to join. Or if Rory, maybe you're in control of that still. All right. So I'm going to talk about some of the work we've been doing over the past 5 years, mostly in the west coast of Ireland, but some of it throughout the whole coastline, including parts of Northern Ireland. And this is a collaboration between researchers in NUIG, University of Alabama at Birmingham, Search, and we feed a lot of our information into the National Biodiversity Data Center and work a bit with Dave Walls on the Shore Watch project. So I'm just going to start, I think, by talking about kelp forests. So these are marine forests. And that term is used pretty loosely to describe a lot of different algal communities, but I'm going to be talking about actual forests that are kind of comparable to what you find on land. And they're found throughout 6 continents of the world. None of them are found on Antarctica, and that's largely because the temperatures are too cold for the sporophyte, or the more apparent form of the kelps themselves. In Ireland, we have mostly Laminaria species. These are a shorter kelp in comparison to the giant kelp that you'd find in the North Pacific, South Pacific, or the Nereocystis or bull kelp found in the North Pacific. But they form really dense communities that are really important for marine life. Now, these communities are pretty threatened by a variety of things. They can be biological stressors like overgrazing by urchin communities. And this is a pretty rough picture I pulled from the internet. It's a picture from Norway, where the purple urchins can kind of decimate whole swaths of Laminaria hyperborea communities periodically, especially in the north coast of Norway. We don't really have that kind of grazing pressure in Ireland, but it is something that could potentially happen if we were to get these kind of ravenous herbivores in our systems. We also have climate change, and that is causing ocean warming in the North Atlantic as well as other regions of the world. So this is particularly detrimental for cold water kelps, which are experiencing sort of a northward retreat or a poleward retreat in their communities. So they don't really respond well to warm waters and to warm high summer temperatures and such, and may be replaced by more warm water kelps that you might find in Europe, such as Laminaria ochrolouca, which we also call the golden kelp. So Laminaria hyperborea is fairly long-lived. I've seen a 15-year-old in Ireland, generally between 10 and 15 years maximum in communities, but they often are taken out by storm activity and such before they reach that age. Has seasonal reproductive patterns, so it becomes reproductive, or you see these dark patches on the blades called sori from October to February, and these produce zoospores, which then turn into the gametophyte stage, which I'll talk about a little bit more later in the talk. It also has seasonal productivity patterns, so growth and biomass creation. And these generally growth will start occurring from January through May. And you can see here, this is kind of a picture of kelps, different kelp stages you have. I don't know if you guys can see my cursor, but you have kind of a normal adult that you see in the summer, a larger adult that, or kelp that you will see growing from February. So you see this bulb at the base of the blade, and that's the New Year's growth, whereas the lamina that's at the very end of the blade is last year's growth, and that's shed around May. And that would have had all the reproductive structures and such from before. This is a very important marine habitat in the context of the things that are growing on them or that live within the forest themselves. And again, its distribution is predicted to change due to climate shifts. So this is kind of a nice graphic from Brody et al. In 2014. And you can see that in our boreal region of Europe, we're predicted to see a shift to less calcified species like marl and more warm water kelps like the golden kelp or invasive species like undaria. Whereas in the Arctic, you'll see a shift towards more kelp cover, including Laminaria and Saccharina latissima. So that's an issue, but in the past, we know that Laminaria hyperborea communities have shifted. So with the Little Ice Age, these communities were pushed towards the equator. And then as the climate naturally warmed, we saw these communities expand their range up towards Svalbard and northern Norway. And actually, our region of Europe is predicted to be a glacial refugia. So that means that kelps persisted through ice ages in this area and may harbor unique genetic diversity that's important for the species' persistence or resilience. And I'll talk a little bit about that later. So, kelp forest ecology is pretty well known worldwide, especially in certain regions. So where I come from, Central California, there's a pretty good understanding of kelp food webs, and it's depicted by this graphic. And this kind of data is really boosted by different kelp forest research networks. And these are just some examples here below of networks worldwide. So you have PISCO, which is in North America, NIVA, which is the Norwegian version of this, Keen is an international partnership where people contribute data from all different research areas, and MARFOR, which was a biodiversity project in Europe that was particularly run by, I think, Germany, France, and Portugal. And again, we're trying to do this biodiversity. We've applied for a biodiversity grant again to kind of create a kelp forest or seagrass network in Europe and kind of boost our understanding of how these ecosystems work. But what I want to say, coming from this graphic is that we actually don't really understand food webs in our Laminaria hyperborea communities, especially in Ireland. What we do know is really drawn from work that's been done in Norway and France. And that'd be studies on isotope levels within different trophic communities and really looking at gut content of birds and other top predators and so on down the food chain. But we haven't really done anything like that specifically in Ireland. And Ireland has a pretty unique position in the Northeast Atlantic, kind of taking the brunt for most of the Lusitanian region of Europe. So what we wanted to do about starting 5 years ago is create a sort of a modern baseline for kelp forest ecology in Ireland, really just understanding how productive these systems are in multiple ways, but also how diverse they are, especially in comparison to other ecosystems in the nearshore environment. And that would be things like marl bed or saprolid worm reefs. We also aim to try and create a monitoring program that can contribute to worldwide networks, and that would be something like KEEN, Kelp Forest Ecology Ecosystem Network. And so far, we've really only been able to do this during the summer at one site, but just kind of creating tools that people can use for monitoring in the future, whether they be remote sensing tools or things like swaths that scuba divers can use. And then lastly, we really want to understand the resilience of these habitats across the coastline. And I'll really dig into what I mean by resilience in a little bit. But first, to really create a baseline for kelp forest ecology, we had to set up monitoring sites that we can maintain for about 2 years. And what we did is we put out monitoring buoys that were subsurface, so they weren't picked up by fishermen. unfortunately, they still were picked up by fishermen, and they also were removed by storms, tried as best as possible. To these, we attach data loggers that can record temperature and light levels in the environment, both in the subsurface, so within the kelp canopy, and at the base of the buoy. So that's kind of like your immediate downwelling light. And we also attach recruitment habitats to look at what the pelagic stock of recruits, mostly like things like invertebrates or fish, were, but also at the base to look at what's recruiting within the kelp forest, so from adult organisms within the kelp forest, or selectively recruiting from the pelagic cohort into the kelp forest. So we had things like a tuffy, which is basically like a dish sponge, but it's kind of got a nice intricate kind of matrix for things to settle out in and hide from predators. But we also had these larger ones that are really useful for pelagic fish recruits, and actually captured quite a few more organisms than the smaller types. We also laid out succession plots, and this is to look at how communities recover after a disturbance. So for instance, a big storm activity. This is Bridges of Ross, this picture right here. So the main thing you'd have here is these big boulders that used to be the old bridges kind of turning over in large swell events. And so we look at the recruitment of new organisms and the growth of kelps and stuff like that in those plots. To look at biodiversity of the habitats, we did swath counts. So we counted basically everything within the 30 by 1 meter transects at 3 locations within each kelp forest every season. So this captures basically organisms that are there year round, but also organisms that are seasonal within the habitats. And we did biomass assessments at 3 different depths within these communities. And that allows us to look at productivity in multiple ways by bringing these plants back into the lab. And then lastly, we did growth plots. And this is basically something that you can do with like grasses, seagrasses, with kelps. You basically punch a hole in the blade and measure how far it grows over a certain day length. And that gives you an idea of how fast each individuals grow. And you can actually do this by age grouping, which is something we just reanalyzed at one of our continually monitored sites in Connemara. So I'm just going to briefly go through the data. And I mentioned before that there's seasonal productivity within these organisms. And if you look at linear extension, it generally, it peaks kind of late February to March. So that's the growth of the blade. And the growth is something around 1.5 centimeters a day, which is quite a lot, actually. It's not as much as what you'd see in giant kelp that grow about 1 meter a day in their peak performance, but it is quite a lot considering how dense these kelp forests are. We also look at pigments, and these are the compounds that all plants and marine algae, or all algae, use to capture light energy. So you have different pigments, predominantly chlorophyll A, which is the center of your photosystem, but also accessory pigments that for brown algae will be chlorophyll C, the carotenoids, the xanthophylls, things that you might hear about when you when you're looking at maybe holistic medicines and reactive oxygen scavenging compounds, things like, you know, they'll give you blueberry extract or, you know, algal extracts and stuff like that in these pills and stuff. And that's what these are. They're basically just pigments that can capture light energy or protect the photosystem from harmful UV rays. And so what we really wanted to do when we were looking at this is look at shifts in pigment concentrations over time in these kelps and get an idea of whether they move with biomass creation or so on and so forth. So what we found is that they do, that in spring when you have the highest growth, you also have the highest chlorophyll C2 and fucoxanthin concentrations. And you can see that these kind of decrease through summer, autumn into winter. And so these pigments are trending with biomass creation. And that's just kind of an interesting fact, there are seaweeds, for instance, in Antarctica that will elaborate these kind of pigment compounds prior to the onset of high light conditions and anticipate kind of seasonal fluctuations in the environment. But what we have here are species that really respond to increase in light conditions. So that's just something that's kind of cool. We can track how the infrastructure of the kelp respond to the environment and growth rates. Now, what's interesting from a blue carbon perspective or carbon capture point of view is the biomass of kelps. And so what we have here is the number of kelps per meter squared. This is kind of the data that I'm presenting to you. And we have your average age in these meter square plots and the density, and you can see that these kind of trend with depth. So density doesn't necessarily trend with depth, but age does. So it tends to decrease as you get deeper, and that just means that these communities at deeper depths have higher turnover rates. They're more disturbed maybe than the shallower depths. And we also see that as you have denser communities, and note again that this didn't really trend with depth, and you have younger median age in that community. So what this means is that when you have something like maybe 70 kelps per meter squared, these are all very young kelps. And as they get older, those communities start thinning. And so it's something like what you would see in terrestrial forests, right? You start walking through a forest, you see a young saplings on the edge of the forest. And as you get to the deep forest, most of the individuals are older, bigger, and the subcanopy community is composed of things like ferns and such, which is kind of comparable to the epiphytes and all the other seaweeds, the subcanopy seaweeds that you see in kelp forests. So these species self-thin. It's pretty interesting in terms of applying an applied perspective on kelp forests. For instance, if you were to harvest or try and help these systems recover after a major disturbance. We also are looking at carbohydrate compounds. So these are compounds that are pretty important for not only energy storage in kelps, but also for their consumers. And you can see some interesting trends in these compounds. So laminarin and mannitol are your major storage compounds. You see an increase in laminarin in winter and spring and a corresponding decrease in mannitol there. You'll also see that this is site, it depends on the site, so the location that you're collecting the kelps from. That's kind of interesting because they have different age groupings depending on their exposure level and the site, just the site itself, site-specific variation. And if we look at the different age groups, sorry, you'll see that once you get to the median age for kelps in Ireland, 3 through 5 or 6, the laminarin concentrations increase significantly. Mannitol generally stays the same until you get to up to 9-year-old kelps. So you just see some very interesting patterns, and this would be something, again, that could be useful in an applied sense if you're targeting these compounds for harvesting or looking at food web perspectives. Okay, so now I'm going to kind of talk a little bit about biodiversity. That was mostly productivity. When we're looking at kelp forests in Ireland, we really wanted to compare them to other habitats. So that included sediment bottom habitats, for instance, in Kilkieran Bay, bedrock habitats, so bedrock walls, and marl beds and sapurlid worm reefs. And so this is kind of a map of all the different sites from Lough Ine through Cork, Kerry, and then up to basically just Belmullet is where we did these studies. But we also have our monitoring sites that are largely in Cork and Galway, again, the west of Ireland. and those are monitored seasonally. So the way we looked at biodiversity is we use a swath survey. Basically, again, it's 30 meters along a transect, and you count all the individuals, the organisms that you find within 1 meter of that transect line. And what we got back was some pretty interesting data. So we do these studies in kelp forest, again, seasonally, but at all the other nearshore communities is opportunistically. We did this with Sea Search, and we go out and do these swaths whenever we could possibly do it. So here we have our symbol is the habitat type, and then the little the label is the season, and you can see that kelp forests pretty much just cluster together despite season, and so they're pretty significantly different from all the other habitats. You see Merrill kind of clusters together, and so does superb worm reefs and such. But if you look at the individual sites, our 4 monitoring sites, they actually are pretty different from each other. So while kelp forests are a unique nearshore habitat, they also have incredible variance in their biological communities depending on where they are on the shoreline. And so that can be kind of maybe muddle the water a bit if you're trying to do a conservation or maybe inform MPAs. But it also just highlights that, you know, these communities are incredibly important no matter where they are. When we do an analysis trying to pick out key species that define kelp forests, what we find is that the echinoderms are really what pop out. So if we look at species that you will always find in kelp forests, it's the common urchin, it's the cotton spinner, very small common SARs. You got the spiny sea star as well, and then a lot of gastropods. Obviously, the blue-rayed limpet is very intricately tied to Laminaria hyperborea, and it's really found near the apical meristem in summer, but also these top shells and so on. We also see that there's a lot of commercial species in this habitat, so the common crab or the lobster, and the lobster seems to be very important in regulating urchin communities. But again, I have to stress that these urchin communities, specifically the common urchin, is not a ravenous herbivore or anything like that. It's not going to overgraze these kelp forests. It usually will feed on detritus, not on the live kelp itself. Something else that kind of pops out is that they have very dense juvenile gata communities from late spring through summer. So if you look underneath each of these yellow circles, there's a little baby fish in the canopy there. And you'll often see them kind of feeding on small nudibranchs or detrital carbon that seems to kind of come up with the swell. And theoretically, they might be using this canopy as a refuge from predators, but not a lot of work has really been done on this. There's a study from Norway saying that, yes, seabirds that are associated with kelp forest canopies have a large number of juvenile fish in their stomach contents, but really I think that this is a very interesting aspect of kelp forests that needs to be kind of looked at more in depth. Oh, there you go. But yeah, so no one has ever looked into the use of these kelp forests as a nursery. Okay, so now I'm going to kind of delve into recruitment to kelp forest specifically. Now, this is very tricky because, as I said, kind of in the start, these monitoring buoys, we put them subsurface so that people wouldn't see them necessarily and that they wouldn't get entangled in people's props if they were boating nearby. But they did, like if you had a very low tide, you could potentially see them, and quite a few of them were taken. So we have very mixed data in terms of recruitment. Some seasons we missed out completely on a few sites. And sometimes these things just, the mooring or the recruitment habitats would just be taken by storms as well. But we do see interesting patterns in location for Tuffy recruitment. Again, this is the smaller recruitment habitat. And you have your season in the symbol, and then the acronym here just means different sites. So you see some seasonal separation, spring and summer and autumn in the types of the communities that are found in these Tuffies, but also a little bit of site separation And that would make sense, especially if you have a pelagic larval cohort, because County Clare and County Galway, where our monitoring sites are located, have different current systems kind of moving around them. The larger habitats, which we call Smurfs, captured a lot more organisms actually, including the small species that we were expecting to find in the toughies, but we lost them a lot more frequently. So you don't get this nice pattern of site or seasonal clustering. But we do see that, you know, especially in springtime, we are getting a lot more of the crustaceans. So from maybe your intertidal crab species all the way to the common crab and the lobster and the squat lobster species. And we're getting a lot more gastropods later in summer and so on. So we see some small patterns, but really it would be nice to kind of keep repeating this and compare recruitment in different habitats to really highlight how important kelp systems are. We had these nice habitats, you know, in the pelagic region of the kelp forest and then underneath the kelp canopy, but you can't really compare them when you lose so many samples. So that'd be something nicer to pick out as well in the future. Now, I also mentioned that kelps themselves are an important habitat. And so this is just a rough graph of site biomass with diversity and species richness. And you can see as the kelp ages or becomes bigger, and the communities become more diverse and richer in terms of the number of species inhabiting them. And these are mostly sessile species, so tunicates, sponges, epiphyte communities. And the same pattern is found in the holdfast. So every year as the blade grows, the kelp is laying down a new set of haptera. And so as the kelp gets older, the communities get more diverse. So it's pretty cool. And we think that we found a rough estimate of the richness in kelp forest communities is around 320 species in total. And this is ignoring the diversity of the Nemertians. We really needed a worm specialist to kind of help us with that sort of stuff. But yeah, much more diverse than what the other habitats we investigated. Now, something interesting that we found in terms of succession or recruitment or recovery from a disturbance, we cleared native plots, so leaving coral and algae and the turfs in the community because we weren't about to bleach or, you know, blowtorch subtidal habitats. We really just wanted to look at how the community recovered after the canopy was removed and other sessile organisms. But we also put in 1/4 meter squared cement blocks to see if these kind of artificial reef or succession plots would recover in the same way. So really what we're looking for is potentially a microscopic community in the native succession plots that will facilitate recovery more than artificial reef might. So with the native plots in each habitat, what you see is fast kelp growth. These were cleared in spring, just so you know. So when the storm events are pretty intense. Kelp growth over 101 days. By July, you start seeing thinning in these communities, although the animation's not working very well. So these communities self-thin as these kelps get bigger, which is something that we saw just in our plot clearance. And that's about 208 days. And then after a year, these plots are basically not recognizable. So the adults kind of take over. On your sterile succession plots, what you see is kind of a bacterial turf community forming, which captures sediment in it. That's after 41 days. You start seeing settlement of calcified invertebrates. So this is spiral bed worms here. And then you get sponges and tunicates coming in after 148 days. And then after a year, presumably after a reproductive event in the adult kelps in the community, you start seeing growth of juvenile kelps. So really you need that plot to be conditioned by the marine environment, and then you need a reproductive event. So theoretically on these coral and algae these native succession plots, these kelp spores were already present, or gametophytes or juvenile sporophytes were already present in facilitating recovery in that habitat. And we have a few replicate experiments underway in Connemara. So quickly, I'm just going to talk about kelp life cycles. So you have your very apparent, what we call sporophyte. This has 2 chromosome pairs, quite like us. It produces zoospores when it's reproducing. So when the sori form on the blade from October through February, it's producing zoospores, which are released. You get higher numbers released later on in winter, so January, February. Those settle out in the benthic boundary layers, so on boulders, coral and algae, whatnot, and they develop into either male and female gametophytes, and those are quite like ovaries and testes that you find in mammals. And these have only 1 chromosome pair. So both sets of gametophytes produce gametes, so an egg and a sperm theoretically. The egg exudes pheromones quite like mammals do that attract sperm, and then once those recombine, they form a juvenile sporophyte, which grows into this large apparent adult sporophyte that we can see and what forms the kelp forest. So we're really interested now in what happens directly after zoospore settlement. So do gametophytes develop immediately? Can they lay dormant for a period of time until they decide it's optimal to produce eggs and sperm? Do they do that right away? And does the sporophyte remain dormant when it's only 1 to 2 cells big? Lots of different options theoretically might happen in the benthic boundary layer here. And so that's our focus of research here now. And so we're looking at resilience in kelp ecosystems, defined as the capacity of these ecosystems to recover from a disturbance and maintain normal ecological function. So house the same amount of biodiversity and maintain the same level of productivity. And we want to know this because there are so many stressors. So you have your short-term stressors like marine heat waves, kelp removal, annual storms, but also long-term things like climate change, which may mean warming, but in the long term for Europe actually could mean water cooling, depending on how the ice sheets respond to climate change. It could also mean frequent storms and maybe the onset of mechanical harvesting across the coastline of Ireland. I mean, this could be particularly negative for kelps if they're stressed by climate change as well. So I showed you this earlier in the talk. This is just a nice graphic showing you the extent of Laminaria hyperborea forests in Europe from the last ice age to present day and then to future predictions. So with warming, we're expecting a poleward retreat. So these species will move northward because they can't handle the warm temperatures, and they'll be replaced by warmer water kelps like Laminaria ochreluca. So to kind of target resilience in kelp forests, we really wanted to know where these habitats have been historically. So using herbarium or long-term data sets that you can find on OBIS or GBIF, National Biodiversity Data Center. We're trying to look at where kelps have been recorded in the past, present day. We want to know what the genetic structure and connectivity across habitats is in Ireland. So where do you find the highest genetic diversity, and how are kelps kind of basically connecting across populations? So if you think about the zoospore stage, which I talked about a little bit earlier, these are mobile, like sperm, but they don't swim very far on their own. So potentially they could be carried by currents, or the adult could be detached and moved by swell or storm events. But theoretically, they're only really dispersing about 200 meters. So how are these populations connected, and how can you maintain genetic diversity across the coastline or not? And then we also want to know, is there natural resilience in the life cycle? Again, this looks at dormancy potentially in the gametophyte or the juvenile sporophyte stages in these habitats. And moving forward, how can we continue to monitor these habitats and look at the resilience in the community? So whether you're using remote sensing, again, or scuba diving surveys to watch, to kind of map shifts in populations or a breakdown of the biodiversity in these habitats. And then again, that feeds into historical records of Ireland moving forward. So what we've found is that it's very hard to find good records of subtidal species, especially bigger kelps. So very few of them are recorded in herbaria because they're very big and juicy, potentially moldy if you're trying to press them. And also people in the past haven't really been doing subtidal collections, right? Because things like scuba or even the diving helmet are about a century old. So there wasn't a lot of opportunity to do these deeper water collections. The earliest kelp forest we have on record is actually on Hookhead, 1913, which is quite cool. But we've also noticed that even though we have this kind of early record, the next earliest are up here, Clare Island and such, sites are rarely repeatedly sampled. So if somebody's going diving maybe when they're 20 to Hookhead, they're not then reporting that kelp forest in subsequent years of diving the same sites. So you don't have a good record of shifts in population in that sense. You have the earliest record of that, of the site being visited. So we found no more than 6 years of recording a kelp presence at any given site. And then the next thing is that we don't really see ecosystems being recorded. So there's no mention of how many kelp are found at one site or the state of the kelp, whether the kelp was found as drift, washed up on the beach, which theoretically means it's found nearby underwater, or if it was seen while snorkeling or diving. So a lot of this kind of historical information is kind of fragile in the sense that we can't really interpret too much from this information. Oops, sorry. But you do, so what we need to kind of do moving forward is kind of create an ecosystem, a template for people to record ecosystem health and things like density and abundance. Now, if you look at genetic diversity of habitats, what we've done in the past 2 years is sample habitats all over the coastline, but we've already done an analysis of 8 sites. So from Lough Ine to Zetland Pier to County Clare, Galway, and St. John's Point. And we've looked at the diversity of these different sites. So we take 30 individuals, we extract their DNA, and we amplify certain microsatellites. And we found that the highest abundance is actually down in the south of Ireland, which was kind of predicted because this is theoretically a glacial refugia from the last ice age. But we've also found that Ireland has quite a lot of genetic structure, or is pretty unique when you compare it to French populations, for instance, in Brittany and Portuguese sites. And so this is only 8 sites worth of data, but we have now 48 sites to kind of look at, which can hopefully inform conservation and potential, other users of these habitats. But we've also expanded our analysis of diversity of Laminaria hyperborea in the EU. And so there should be some new data coming out just highlighting how important Ireland is for this species in terms of genetic diversity. This kind of sampling across the coastline has also allowed us to find non-native species. So things like Undaria pinnatophyta, which we just, Keenan, Chan, and Tony O'Callaghan found in Greystones. But also Laminaria opaluca, which is found in Belmont Peninsula. And in 2016, somebody actually did a habitat suitability model for the coast of Ireland in the UK. And this species is kind of predicted to be on a lot of the offshore islands in Mayo, Galway, and also down in Clare and Kerry. So I just think it'd be nice for people to keep their eyes out if they get the opportunity to go to any of these offshore sites, just to see if you see anything that looks like this species here. So it looks like hyperborea, but smooth stipe and very few epiphytes or epibionts growing on it. So again, the south of Ireland is the most unique. You have isolation by distance within these populations, and what that means is that they're not dispersing very far, so there's not a lot of genetic connectivity within populations, which you might expect. And then we have a lot of data kind of pending. Sorry. To look at ecological resilience or resilience within the microscopic life cycle stage of these kelps, we kind of have taken a three-pronged approach. So for the past year and a half, up until September last year, we had a big experiment in two locations, so in Galway and in Kerry, Derrynnon. And what this was basically a cement block that we put in place with microscope slides attached to it, some that were left in situ for about a year, some that were replaced seasonally. And these theoretically captured recruitment of kelps as well as other organisms over a year or seasonally. So that gives us an idea of throughout the year of whether or not kelps can persist longer than the season that they settle in. But also, sorry, hold on a second. Longer than the season they settle in, but also how often they're recruiting, whether there are other species of kelp recruiting. And this was kind of put in place across a gradient within the kelp forest. So from the inner kelp forest to the outer edge and at multiple depths at both sites. We also use metabarcoding of both bedrock and these microscope slides to examine the genetic diversity and kelp species that are settling out, because like many things, kelps, when they're in this microscopic state, are not very easy to identify to species level. We've also been able to grow kelp in aquarium, so take benthic samples into the lab and look at what grows out of it. So that could be anything from red algae to kelps to smaller invertebrates, but it's provided a lot of interesting data in terms of what's present in the rock at the microscopic state, so what these spore banks contain. And so a lot of that data is still pending, but hopefully we can present it next time we have a talk here. So just so you can see, this is what the zoospore stage looks like. This is a video that Keenan took right after they settle. They've lost a lot of their flagella and they're just kind of moving around. And then this is what the gametophyte stage looks like here. This is a 2-cell size thing. So you're just getting an idea of how small these things are and how hard they are to identify in the system. Again, we see higher densities of zoospore coming out in late winter, so January through February. And you see, theoretically, these are supposed to settle within 24 hours, but in lab, they still move after quite some time. And then you see development of gametophytes and then slight thinning of the population in those conditions. And this is just kind of a brief movie here, showing you what the slides look like in situ. So you have your annual sites here. You can see there are quite a few kelps growing on the slides, but also on cement. And then this is your seasonal slides here that we would bring into the lab. So a lot of that data is still pending, but that's kind of how we're targeting those research questions. And I'm going to end on how we continue monitoring kelp forests. So I've mentioned dive slates for scuba divers, for recreational, for clubs, if they have an adopt a site program. You can kind of look at, take down information on the density of the different kelps in the habitat, as well as maybe some of the key species. So these are a lot of echinoderms. This is the QR code to access that, that sheet online, or we can send you waterproof, waterproof dive slates if you need. We also are working with somebody in maths and statistics, Aaron Golden, and he's created a rough metric for looking at kelp forest distribution on the coastline using the red-green, sorry, I'm losing my words, the red-green light information spectra. And so if you take the red by the green, that's the light that's kind of pushed back from kelp reflectance. You can get an idea of how abundant they are down to about 10 meters depth. So that's here, this is kind of your best effort. If you go from lowest astronomical tide to 10 meters, you're getting mostly hyperborea, but if you go to plus 5 meters, 10 meters, you're getting all the kelps, including digitata. If you go down to 15 meters, you get a lot of backscatter, okay? So that has to do with the downwelling light just kind of refracting within the water column. I'm using the wrong words here, sorry, but basically his tool, and hopefully we can kind of create a template for people to do this on their own, is pretty useful from the intertidal down to 10 meters depth. And that's fairly good for kelp forests that are found along the coastline of Ireland. They get much deeper on their offshore islands, but where you have a lot of nutrient runoff or terrestrial runoff, they really do go down to about 10, 15 meters max. And we also have kind of these flyers. It's kind of a new thing for people to help us record where they see different kelps. So for instance, in Greystones, Undaria has probably been there for quite a while, but it's just hard to distinguish from other kelps. So we're hoping that people will kind of take these cards and kind of use them to ID kelp species that they see on their dives and help us record that information in our online form. And so I'll leave you with this. And moving forward, I hope that we can kind of create tools for people to monitor kelp ecosystems. And we should be able to really define resilience as we wrap this project up. This is an EPA-funded project that CSearch is part of. That ends in October. So hopefully there'll be more data as well as an EPA report on kelp forest kind of coming forward. And then if you look at this link down here, this is pretty important, I'd say. There's a GoFundMe to help CSearch kind of get money to support their underwater monitoring. And I know they're doing this in Sapperlid worm reefs right now. And this is a fairly understudied habitat. So if you're feeling flush with cash, go to this site right here and kind of help them maintain the work that they're already doing, their own goodwill. Okay, so that's it for my talk right now, and I'd be happy to take any questions for the next 10 minutes. I kind of have to leave in 10 minutes, so should I give it to you, Rory?
Rory might have skipped off to put a young lad to bed. So I'm not seeing any questions in the chat room at the moment.